to the another video tutorial from DI Lectures. In this video lecture, we'll be discussing about the signal transduction mechanism, right? So, we know that जो भी सेल हमारी बॉडी में होते हैं, it has to synthesize certain important kinds of proteins, it has to synthesize certain important kinds of enzymes. And in this video lecture, we'll be discussing about how those enzymes and proteins are synthesized and how the signal from the outside cell is being transduced into the interior of the cell, and that interaction between the outside signal and the plasma membrane is being transduced into the synthesis of certain kinds of proteins and certain kinds of enzymes or any other important kinds of compounds what a cell needs, right? So, for example, if we draw a rough diagram of a cell here, right? So this cell has certain, we know important kinds of cell organelles are present inside. It has a nucleus present inside, and nucleus contains the genetic material, right? It has endoplasmic reticulum, it has Golgi complex, it has mitochondria. Different kinds of cell organelles are present. So if this cell is running low on certain kinds of proteins or enzymes, right? So जो signal है, that will come from outside. Signal बाहर से आएगा because signal is present outside in the extracellular fluid, and that will come and will show interaction with the outer boundary of the cell, what's called as plasma membrane. And that interaction between that signal and the plasma membrane will be transduced into the interior of the cell, and from the interior of the cell, then from the cytoplasm, that signal will go to the nucleus and will influence certain important kinds of genes present inside the nucleus, right? So how we will define the signal transduction? We can say that the specific interaction between the ligand, because जो भी signal बाहर से आएगा, we call that ligand. So we can say that the specific interaction between the ligand which is present outside the cell and the receptor, that means the ligand will come and will show interaction with certain important kinds of receptors present on the plasma membrane. That interaction between that ligand and the receptor will ultimately change the behavior of the cell, will ultimately change the gene expression of the cell, and that is called as signal transduction, right? So in this video lecture, we'll see that how the cell maintains those different kinds of signals. अगर signal बाहर है, तो वो सिग्नल कैसे सेल को इन्फ्लुएंस करती है, कैसे सेल के ब्लूप्रिंट को और जेनेटिक मटेरियल को इन्फ्लुएंस करके अपना काम करते हैं, जैसे कैसे प्रोटीन्स को सिंथेसाइज करते हैं, कैसे एंजाइम्स को सिंथेसाइज करते हैं, राइट? So we know that जो भी प्लाज्मा मेम्ब्रेन है, we made here a rough diagram of a cell. जो भी प्लाज्मा मेम्ब्रेन है, प्लाज्मा मेम्ब्रेन has tremendous amount of these receptors present outside on its cell surface, right? So for example, and every receptor, जो भी receptor है, that will show interaction only with its specific ligand. Got the point? So the ligand will come, that will show interaction with this cognate receptor, and that interaction will go inside and will activate certain kinds of proteins, and those proteins will activate downstream proteins, and ultimately those downstream proteins will go inside the nucleus, and once they reach the nucleus, they will influence or will show interaction with certain portion of the genetic material and will transcribe mRNA, and once the mRNA is formed, mRNA will come from the nuclear pores into the cytoplasm and will form those important kinds of proteins or enzymes जिसके लिए सिग्नल बाहर से आएगा, राइट? नाउ इस हम अब ये देखेंगे कि जो ये रिसेप्टर है, रिसेप्टर जो प्लाज्मा मेम्ब्रेन पे है, ये कौन सा रिसेप्टर होता है, क्योंकि नंबर ऑफ रिसेप्टर्स आर प्रेजेंट ऑन द प्लाज्मा मेम्ब्रेन, लेकिन जो स्पेशल काइंड्स ऑफ रिसेप्टर्स होते हैं विच आर स्पेशली मेंट फॉर दिस सिग्नल ट्रांसडक्शन कास्केड, विल सी वो वाले रिसेप्टर कौन से होते हैं और वो वाले रिसेप्टर किन किन कंपाउंड से बने रहते हैं, राइट? नाउ हियर यू कैन सी द डायग्राम हियर, एंड दिस इज द डायग्राम ऑफ G protein coupled receptor. This is diagram of G protein couplet receptor. G protein couplet receptor. Right? That means here you can see this is the plasma membrane, and this receptor is present on the plasma membrane, and we call this receptor G protein couplet receptor. It's a big name, right? Why? Because we call it G protein couplet receptor. That means it is receptor, but it always and only works when it is coupled with a G protein. You got my point? So simply we can say this is a receptor, as the normal receptors are present on the plasma membrane. But this receptor only works when it combines with certain important G protein. That's why we call it G protein coupled receptor. That kind of receptor only works when it is coupled or linked to important kinds of G protein. Right? So जो G protein होता है, we have multiple kinds of G proteins. लेकिन here what kind of G proteins we'll discuss? We'll discuss heterotrimeric G protein. We'll discuss heterotrimeric G protein. Heterotrimeric G protein. As the name indicates, heterotrimeric. That means 3 different kinds of subunits are there in a G protein. And this receptor, जो यहाँ पे receptor which is जिसको हमने green color में बनाया है, ये जो receptor है, it has how many segments are there, how many fragments of this receptor are there? 1, 2, 3, 4, 5, 6, 7. That means this receptor traverses the membrane 7 times. So we can call it 7 transmembrane receptor. Or we can call it by another name, serpentine receptor, right? We can call it serpentine receptor, or we can call it 7 transmembrane receptor. That means that kind of receptor which traverses the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. Right? This is normal kind of receptor. And जो ये 7 transmembrane receptor है, it has 2 important kinds of loops. One loop is present outside and another loop is present inside. Outside means that is present into the extracellular fluid, and another loop is present inside the cytoplasm. Right? जो extracellular fluid की तरफ जो इसका जो portion present है, that is ligand binding portion. ये वाला portion you can see here, that's ligand binding portion. That means यही वाला वो portion है, यही वाला वो region है इस 7 transmembrane receptor का, which shows interaction with different kinds of signals, which shows interaction with different kinds of ligand which is present in the extracellular fluid. Right? Now, जो inside वाला इसका loop होता है, which is called a cytoplasmic loop. That cytoplasmic loop shows interaction with this heterotrimeric G protein. You got my point? So here, if we conclude, we can say that जो भी signal बाहर से आएगा, that will show interaction with this transmembrane receptor, 7 transmembrane receptor. लेकिन कहां से? बाहर से, क्योंकि ligand binding region बाहर present होता है, और जो अंदर वाला इसका portion cytoplasm की तरफ present होता है, वो interaction किसके साथ शो करता है? G protein, heterotrimeric G protein के साथ, right? और यही हम अभी बता रहे थे कि जो बाहर से सिग्नल आएगा, that will show interaction with this kind of receptor. You mean this receptor is a 7 transmembrane receptor, और ligand बाहर से आएगा. Once this interaction between the ligand and this G protein coupled receptor, that interaction, that signal will be transduced inside the cytoplasm by different kinds of signal transaction cascades or events. We'll discuss them in later lectures, right? And ultimately signal will go to the nucleus and will influence certain region of genetic material, and जो भी प्रोटीन उसको बनाना होगा या जो भी एंजाइम उसको बनाना होगा, एंड वो बनाया जाएगा, क्योंकि जब ये इन्फ्लुएंस करेगा उस पोर्शन, जेनेटिक मटेरियल के पोर्शन को, और वो mRNA बना के उस एंजाइम और प्रोटीन को बनाएगा. Right? Now, in this video lecture, हमने ये देखा कि जो G protein coupled receptor है, this is simple a receptor which is present on the plasma membrane of every cell, and this receptor traverses the membrane 7 times. We call it 7 transmembrane receptor, and or we can call it by another name the serpentine receptor, and after this once it will traverse the membrane, then it will show interaction with the ligand outside, right, and ultimately जो इसका अंदर वाला पोर्शन रीजन होता है वो इंटरेक्शन किसके साथ शो करता है, जी प्रोटीन के साथ. You got my point? Now, by this interaction, different kinds of enzymes, different kinds of proteins are synthesized, जिसके लिए सिग्नल बाहर से आके सेल को इन्फ्लुएंस करेगा. You got my point? The signal जो भी बाहर से होगा, ligand होगा, it will be any kind of nucleotide, it will be any kind of peptide, it will be any kind of nucleosides, or it will be nucleotides, it will be peptides, it will be certain important kinds of amino acids. So all those will act as a ligand, then they will interact with this G protein coupled receptor and they will do their work. Right? So in the next lecture, we will discuss in detail how that signal is being transduced and how those G proteins are activated and how the signal is then ultimately transduced to the interior of the cell, that means to the nucleus. Thank you.